Boa tarde a todos. É com muita satisfação que hoje, dia 1º de abril, estamos abrindo a série de webinários do Centro de Desenvolvimento Tecnológico em Saúde da Fiocruz, do ano de 2021. Os webinários do CDTS, eles ocorrem usualmente na última quinta-feira do mês, às 10 horas da manhã, podendo haver alguns ajustes de acordo com as agendas dos nossos convidados. Antes de iniciarmos, eu gostaria de esclarecer que o webinário de hoje vai ser ministrado em inglês pelo nosso convidado especial, Dr. Austin, e as perguntas e comentários, elas podem ser realizadas via caixa de comentários no YouTube, em inglês ou português. Esse seminário, bem como todos os demais da série de webinários do CDTS, vão estar disponibilizados no nosso canal do YouTube, CDTS Fiocruz, após essa sessão, onde você também pode encontrar os seminários do ano de 2020 lá gravados. E para completar, eu gostaria de agradecer imensamente a presença de todos. E a partir de agora, eu vou começar a minha comunicação em inglês com o Dr. Austin. Good afternoon, everybody. My name is Poliana Zorzal, and I am pleased to host our session today. So, for our very first session of the CDTS webinar series in 2021, we are delighted to receive Dr. Christopher Austin, the Director of NCATS, National Center for Advancing Translational Sciences, which is one of the 27 institutes and centers at NIH, National Institutes of Health in the U.S. Dr. Austin has served as director of the NCATS since 2012. He founded several initiatives, including the NIH Chemical Genomics Center, the Therapeutics for Rare and Neglected Diseases Program, and the Toxicology in the 21st Century Program. From 2016 to 2018, he served as chair of the International Rare Disease Research Consortium. Dr. Austin is also a member of the National Academy of Medicine in the U.S., and he earned an AB from Princeton University, an MD from Harvard Medical School, and completed training in internal medicine and neurology at Massachusetts General Hospital. Dr. Austin, in my name and in the name of the Center for Technological Development in Health, CDTS, and its director, Dr. Carlos Morel, and the whole Oswaldo Cruz Foundation, Fiocruz, I would like to thank you once again for accepting our invitation and taking the time for kicking off our webinar series. Without any doubt, it's to get off to a great start with you here with us. Your experience after almost a decade leading one of the most developed and important centers related to translational science and medicine in the globe will for sure be of great use for us at CDTS and Fiocruz. Just as a general announcement for our audience, we have to cut short our session today, but please feel free to interact with comments and questions during Dr. Austin's lecture. You can use the YouTube channel chat box to write down your name, institution, email address, and questions, either in Portuguese or in English. Even though we may not have time to address all of them today, we will try to reach out later with answers. I also would like to take the opportunity and say a big thank you to Marco Tulio, who led the connection with Dr. Austin's team, making this lecture possible. We also would like to take the opportunity to thank our support team, which is responsible for the webinar's organization. So without further ado, I would like to give the floor to Dr. Austin. Over to you, Dr. Austin.
Well, thank you, Poliana. It's a great pleasure. It's been a great pleasure to work with you and your team in preparation for this. And you've even taught me a little Portuguese, so I'm learning. And it's really wonderful to be here with you today. We have collaborators at Fiocruz already, and it's great to be able to talk to more of you today. So what I'm going to try to do is give you a very rapid overview of how NCATS thinks about the translational science problem. Talk just a little bit about some of the international collaborations that we're doing, and then tell you how we pivoted to COVID-19 about a year ago. Most of our programs addressed COVID-19, probably much as they did in Brazil there. And it was fitting to do that because COVID-19 illustrated a lot of the general problems which exist in all of translation for any disease. So it just brought them to the public consciousness, I think, in a more widespread way. So I'm going to try to share my slides here, and Poliana, promise to yell at me if I can't be seen or heard. So I'm going to try this and see what happens.
Yeah, you are already sharing your screen.
All right. Poliana, can you see my slides now?
Yeah, it's perfect.
Okay.
It's working.
Okay, perfect. All right. Well, here we go. This is the general problem that I think is the biomedical problem of our era, and that is a divergence between fundamental science and our ability to treat disease. You know, when I was in training almost 40 years ago now, we really didn't have most of the understanding or the tools that we have now. And I've just shown you two of them. On the left side, the human genome sequence, of course, and on the bottom left are induced pluripotent stem cells that are differentiating into neurons and putting out processes. You know, these were things that when I was in training really were science fiction. They are things that graduate students do, or even college or high school students do these days. So a dramatic increase in fundamental basic science to make it almost unrecognizable compared to what it was 30 or 40 years ago. But if you ask on the right side, if you think from the clinical perspective, those of you who are physicians or have been to see a doctor recently, and ask yourself, well, how different is your experience of seeing the doctor today than what it was 30 or 40 years ago? And unfortunately, for most diseases, the answer to that is not very much. It's pretty much the same as it was 30, 40 years ago, despite the amazing understanding in the fundamental domain. And my own field of neurology is one of the best examples of this. Very little has happened therapeutically in neurology in the last 40 years, despite the fact that brain science has progressed by leaps and bounds. So this transition that we all thought would happen by itself if we understood how human beings and biology works in health and disease, there was, at least I thought, and this is what I was taught or led to believe, that treatments sort of just follow as the night follows the day. There would be treatments, but in fact, that could not be further from the truth. We are really quite terrible at going from a fundamental discovery to a clinical intervention that improves health. This process, as I'm sure you all know, is terribly failure-prone, inefficient. In U.S. dollars, developing a new drug costs somewhere between $2 and $3 billion, accounting for failures. The failure rate is well over 95%. And importantly, even at least in this country, and I'm guessing in Brazil, even when there are interventions, drugs or other interventions that are developed, our system takes around another 15 years to get those interventions to all the people who could benefit from them. So going from a fundamental discovery to getting a treatment that gets to all the people that are needed takes around 30 years now. And that has not decreased appreciably. By some measures, it's increased in the last many decades. So we got a big problem here. And sometimes people ask me, well, is translation, this process, new? Of course it's not new. People have been trying to do this, doctors and scientists have been trying to do this for literally thousands of years. What's different is what's on the left side here, this opportunity, the need of patients with disease has always been there, but our understanding has not been. And one hasn't kept up from the other. So that is the translational problem. And again, I think that is the problem of our era. So what is translation? People use this word in many different ways. I have been reminded that, of course, translation is the term we all learned for going from one language to another. You just heard it, going from Portuguese to English and vice versa. And it's also known as the process by which messenger RNA gets made into protein. So when our center, Translational Science Center, got formed, we actually had to come up with a definition of what our field is. And we took a very broad, holistic definition of turning observations into interventions. And that observation could be made in the lab by a scientist. It could be made by a doctor in the clinic. It could be made by a public health worker in the community. And the first time that observation is made, there might be an idea that comes to this person. Well, gosh, if I could just turn this observation backwards, if I could turn it off, maybe the health problem in front of me would go away too. So the whole process from going from that original observation, which may or may not be robust, may or may not be reproducible, into an intervention, a drug, a device, a behavioral intervention, a medical procedure, whatever the modality is, from going from that observation into that intervention and showing that it actually improves the health of individuals and communities. That is the whole process of translation. So it's very broad. So what is translational science then? Translational science is quite different. Translational science is like any other science. It's the field of investigation which seeks to understand what the general principles are by which this process happens. You know, those of us who have been in this field for a long time know that the reason that translation is so terribly failure prone is that we really don't understand very much about most of the steps of this process. It's about a 20-step process to go from a basic observation in the lab to a treatment which is used in the community. And for the most part, each of those steps is empirical. It's trial and error. And there are a lot of reasons for this historically that we could get into if you're interested. But the fact is that this is a trial and error field. It has always been that. And when you think about it on first principles, what we see now in terms of failure rate is exactly what we would expect if we had a complex multi-step process with an increasing number of degrees of freedom as we go from a protein in a tube to a person living in a community. The likelihood of our getting from one end to the other of that 20-step process with an increasing number of degrees of freedom using a trial and error approach, guesswork essentially, that likelihood approaches zero. If you just do the numbers, 20-step process, if you have a 10% likelihood of success, 10% to the 20 gives you, you can do that math, and it's a very low success rate. So the conventional solution to this is just to do more of each one. And high throughput chemical screening is a perfect example of that. The lab that I'm sitting in now at NCATS is a world leader in that field. We are able to test 3 million different chemicals in a biological assay every week, and we do that. And we're very proud of that. However, if you think about it, that is a tacit admission of ignorance, because anyone who has to try 3 million different things to find the thing they're looking for has no idea what they're looking for. And so translational science is the field that hopes to turn that empiricism, having to do it over and over and over again, because we have no idea what the fundamental principles are, into a predictive science. So going from what sometimes feels more like astrology into engineering. And we've seen this happen in a little bit more than my lifetime in genetics, for example, which was a highly empirical field for years and years and years, and is now quite well understood and quite predictive and quite powerful. And that's what needs to happen in translation. Interestingly, there are very few places in the world focused on this definition of translational science. Pretty much every academic organization that I'm aware of, pretty much every biotech, every science, every pharma is more or less trying to get a product out the end for whatever the disease is using pretty much the current state of the art. But that, so what's really needed is an academic field that develops the underlying science that everybody can use. And that's what NCATS was formed to do. Okay, so what exactly do we work on anyway? So, as Poliana mentioned, NCATS the NIH has 27 different institutes and centers. Most of them are research organizations that follow the medical specialties. So there is a neurology institute and a psychiatry institute and a heart institute and a lung institute and a skin institute and a bone institute and an eye institute and a lung and nose institute. You get the picture. NCATS is very different. Our disease, if you will, our organ, if you will, is the translational process. And if the translational process was a patient with a disease, it would be very sick. It would die 99% of the time, and we would have no idea why or how to fix it. So these are the symptoms, or the problem list, if you will, of the translational disease patient. And these are the things that we need to understand and develop solutions for in order to make translation a predictive science. So we start out with the fact, at least in this country, and we found this with our colleagues at Fiocruz so far through one of our collaborations, very few people understand this process of translation. And so that understanding in education is a big issue. And then everything from predictive efficacy and toxicology to data interoperability and clinical criteria and biomarkers and institutional review board harmonization and recruitment of clinical trials and on and on and on and on. You'll notice that none of these have a disease in the name. And that's because the causes of translational failure, going from a fundamental discovery to a treatment that improves health, the reasons for failure, for the most part, are identical, no matter what disease you work on. Doesn't matter whether you work on emphysema or eczema or Alzheimer's disease, the reasons for failure are independent of the disease. And this is another problem with the system. Everyone works from their own disease perspective, and no one works on these problems. And these are the problems that are preventing treatments from getting to patients. And so, but they're not only scientific problems, these are scientific problems, they're also organizational problems and cultural problems. At least in this country, our biomedical research ecosystem is set up to do basic research. All of the systems we have, our universities, our tenure, the idea of a principal investigator, one person who is going to be doing all the discovery, is perfect for basic science. And it's where I got my start, and I love that work. But translation is a team sport. It takes 20 different kinds of people who are very diverse in their fields for success. It requires biologists and chemists and engineers and informaticians and pharmacologists and toxicologists and clinical trial experts and public health people and regulatory people and clinicians. Very, very broad team. And that just does not fit the organizational and cultural model, which is dominant and has been dominant for perfectly good reasons up until now. Because up until, as we started at the beginning, 40 years ago, we didn't know all of this fundamental science. And so, of course, that system was perfect. And it still works great for basic science. It does not work, however, for translation. And so all of these issues, like credit for team science in a university structure, or inclusion of patients on the research team, or education, training, collaborative structures, public-private partnerships, those are all things which are areas of all the things on this list are areas of innovation for NCATS every bit as much as these are. So novel medicinal chemistry and toxicology innovation is, in our mind, no more or less important than solving the incentives for team science problem or the IP problem, intellectual property problem. Okay, so I'm going to show you now some tools and advances I'm hoping will be useful to you. So the first one is addressing this first problem on the problem list. That is that most scientists, physicians, certainly government officials and others do not understand where drugs come from. They seem to believe that they just come out of thin air, their spontaneous generation And they do not, of course. It is a very difficult, arduous process. And so through the National Academy of Medicine, a large group of us from pharma, biotech, FDA, patient groups, universities got together, and we created a map, a realistic map of what it actually took to go from a fundamental discovery to an intervention that improved health. And we published this a couple of years ago, and you can see the references there in two papers. So we started out with this. I hope you've all seen this. This is, you probably all have. It is the conventional diagram, which I have used most of my career to show where interventions come from. This happens to be for small molecule drugs, but there are versions of this for biologics or gene therapy or what have you. But it's basically this model. And it suggests that this process is quite straightforward. It is 6 steps. It is a pipeline. And if you just follow these 6 steps, you're going to have an intervention that improves human health out the end. Even the word pipeline, which we use, I don't know what the term is in Portuguese, but this term drug pipeline is terribly misleading. Because what is a pipeline? A pipeline is something into which an entity, water, oil, whatever it might be, goes in at one end and comes out the other end, unchanged, we hope. And the water better come out unchanged, or else it's a bad pipe. And the progress through the pipe is only dependent on the diameter of the pipe. And it's 100% fidelity. What goes in comes out the end. That could not be further from the truth for the process that we are engaged in. And we realized in this process that these words matter. People believe us when we say there is a pipeline, and then we say this is very difficult, and the public and scientists and physicians understandably say, how can this be hard? It's a pipeline. You just said it was a pipe. So I don't use that term anymore because it is misleading and people believe us. Words matter. So what does it actually look like? Well, this is what it actually looks like. And whenever I show this, I get a combination of laughter, groans, gasps of horror. And this is, like in most scientific publications at the end, this is a schematized, simplified view of each of these. What you're not seeing here is the fact that a lot of these steps, you can spend 2 or 3 years in one step going around and around and around and around and never get out. But fundamental science is at the upper left corner there, and patients are all the way around at the lower left corner. And we use this for a couple of reasons, a couple of these. One is to educate people about how complicated this is. It's not to say don't get into it, but if you're going to go on any long journey, you better have a map and be prepared and know where you're going to go. Otherwise, you will fail. And it is one of the reasons that failure happens. People simply do not know what they're getting themselves into, and they are not prepared. So we also, after we published it, we decided we needed to create an electronic version. I don't think any of us use paper maps anymore. We all use Google Maps or something on our cell phones. And so we created a version of that. So this is on our website, and this is an electronic version that works very much like a Google map does, and I would encourage you to go have a look. And you can start at any one of these points, just like you can within an electronic roadmap. I will warn you that this electronic map has 10,000 different places in it. 10,000 nodes that are connecting all of these. But that really is complicated. If you put a roadmap of the entire country of Brazil or the entire country of the US, which are about the same size, it would be very, very complicated and have tens of thousands of nodes. And that's what this has too. So it also allows us, thinking about engineers, to say, well, what are the problems in this roadmap that are particularly problematic? And if we think of ourselves as translational scientists being systems engineers, road builders, if you will, we want to look at the steps which are most problematic, take the most time, most likely to fail, cost the most money, and those are the steps that we are going to focus on. Use another analogy. If we think of this as a complex, multi-step organic synthesis with an overall poor yield at the end, what we all learned in basic organic chemistry or biochemistry was to identify the rate-limiting steps and develop catalysts to those steps. That is the only way to increase yield. That is exactly what translational science is trying to do for the process of translation. We also, very early on, had to define what a translational scientist is. And a translational scientist is quite different from the geneticist, the very basic model organism geneticist that I was trained to be. I was trained to be a C. elegans geneticist. And the skills I needed to do that, to develop, to look at nervous system development in a worm, are very, very different than what a translational scientist needs. Those people have to certainly be rigorous researchers, just as rigorous, if not more, than the basic researchers, but they also have to be boundary crossers. They have to be team players. They have to be interested in communicating with their other team members, not just their C. elegans. They have to be systems thinkers. They have to think like a team would. If you're on a football team or playing in an orchestra, you constantly have to think about your teammates and compensating and asking, what do they need? I didn't learn how to do that when I was in science. Scientists generally are not trained to do that. But if you are in the translational field, it is essential. And then you have to be a process innovator, as we've been saying. Okay, so, oh no, my slides just stuck. Oh, here we go. Okay, thank you. Okay. Something that you might find, another thing you might find helpful, this is for the preclinical part of therapeutic development. We realized when we started this that there was no, because of the absence traditionally of because of the absence of an academic field of translational science, there was no literature. There was no guide for new people getting into the field to read about it and learn how to do this. So we created this together with colleagues at Eli Lilly, the drug company, to provide detailed rationales and protocols for every step of this process that are robust, reproducible, consistent, high throughput, and translationally relevant. I am of certain age, so I grew up with this. Maybe some of you recognize this. This is how I learned how to clone DNA. This was Tom Manianis's molecular cloning manual. And shortly thereafter, this came out, Current Protocols in Molecular Biology. When molecular biology started, you had to have protocols to understand how to do it. That's what this is, but for the preclinical part of translation. Okay, so how are we organized? And I show you this I show you this for two reasons. One, it's a collaborative organization. This is within the laboratory division of NCATS, so it's about 300 people. It looks very much like a biotech or a pharma company, but it's very, very different too. And though it's within a sort of academic environment, we're also very different there. We don't have tenure. We don't have tenure track. Everyone works in project teams, and every project is a collaboration with someone, some investigator somewhere in the world who is a disease expert or a target expert. And those people get stuck. They're great scientists, but they get stuck at those purple arrows at the top. And they either have a target, or they have an assay, or they have a lead compound, and they don't know what to do. They don't have the experience. They don't have the resources to be able to move forward, and the projects fail. So within NCATS, we have a whole group of collaborative programs where when people come to us to form collaborations, we put them on a joint project team with the people here, and they together move a project down the pipeline here. I showed you, I told you I would never use that word, but I just used it, down this development pathway here. And we want each of those projects to not only have deliverables that are data or leads for further development, or a repurposed drug, or a toxicology profile, but importantly, every study has to tell us how to do this better, has to have a translational science outcome as well. And I'll just show you one example of this. This happens to be under this therapeutics for rare and neglected diseases part. So that's the part that it's operating in, from a probe or lead development up through early clinical trials. And this is a program that collaborates with academic investigators in small companies and sometimes large companies to take projects which are too risky for or not of interest to the private sector, and takes them through to an IND, investigational new drug application with the FDA, which allows you to put this therapeutic into people for the first time. And we have within that organization enormous expertise, and these people are a collaborative team that works on about 25 projects at any one time to get across the valley of death. And I'll show you these are some of the things that the group does on the left side there. For some of you, those words even may be unfamiliar, and that's actually the point. The reason this works is that our collaborators know much more about their target or their disease than we do. They are the disease experts, not us. And they use words we don't understand. But we know the translation, and we know words they don't understand. But when you put those two together, magic happens. And I would urge you, if we have international collaborators all around the world. So if you have projects at Fiocruz which you think would be relevant, contact this person down low. You'll have these slides, and so please do contact him. We do a lot of work on rare diseases. Why is that? Well, for a number of reasons. Rare diseases are a disproportionate cause of morbidity and mortality. Many people, even scientists and physicians, don't realize how many of these diseases there are. There are somewhere between 7,000 and 13,000 rare diseases. We actually don't even know how many there are yet. And what we do know is, however, that though they are, of course, individually rare, they are cumulatively very common. About 8% of the population, which is about the same prevalence as diabetes. Diabetes, at least in this country, is known as a public health problem. It's a public health issue. Rare diseases are not known as a public health issue, even though they are much higher in cost and a much more a larger cause of disability and premature death than diabetes is. And what is even more striking is that only about 5% of these diseases have any regulatorily approved treatment, every FDA approved treatment in this country. And at the current rate of progress, you will be shocked, I'm sure, to learn, unless you've heard me say this before, that at the current rate of progress, it will be 2,000 years before there is a treatment for every one of these diseases. So if anything tells you that we cannot continue doing the same thing, this tells you we need a quantum improvement in efficiency and effectiveness. And one of the ways we're doing this, in addition to understanding the translational process, is to multiplex. Those of us who are biologists and informaticians, you know what I mean by that. We do many things at a time. We don't do one thing at a time. We've never done that in diseases. We consider them separate. They're not separate. They're all connected with each other. There are commonalities among diseases that would allow us to treat multiple diseases, and there are platform technologies for diagnosis and treatment as well. And so we've put together a clinical research network to do these, and I'm going to speed up here because we're going to run out of time. We have developed a platform approach to gene therapy, which is going to allow us to treat multiple diseases at a time. Doesn't really matter what those diseases are, but we're going to be using an AAV9 platform here to develop and make public the full IND enabling process, so that if another investigator wants to use the recipe that we've done, it will be public. FDA will agree that all you have to do is take your gene, put it in this vector, scale up, use the same recipe that we've made public, and we won't make you do all of these other studies, regulatorily required studies. That's ongoing now. That's a great example of the kind of thing that we're trying to do. We're trying to make translational models more effective. We still do 2D assays, the cell culture, cell line assays that we all grew up with, but increasingly we try to go to the right here to include printed tissues and organ on a chip. We have a tissue 3D biofabrication capability, not for regenerative medicine, but rather to develop more complex, physiologically relevant 3D human cell-based systems to test for drugs, and in multiple different organs, with the hopes that what the therapeutics that we discover in those screens will be more predictive of what we see in humans. I hope you followed the tissue chip or microphysiological systems revolution. This is a program that we started at NCATS about 10 years ago, and it has exploded over the last 10 years. If you think about the things that we all had when we were doing tissue culture years ago, we basically had cells in a 2D platform, but we didn't have any of those other things that are in black there. Scaffold, structure, temporal patterning, perfusion, host response, etc. These tissue chips are little miniature bioreactors, multicellular constructs using human cells, which mimic the structure and function of all human tissues. And we use those to study diseases, and we use them to study safety and effectiveness of new drugs. If you haven't kept up with this, please do, because it's a remarkable field. We also have a very large clinical program that works on the clinical parts of that problem list I showed you. It takes care of about 2/3 of the U.S. population. Most of the large academic health centers you've heard of in the United States have one of these CTSA awards. One of the things that we've done here is to innovate on clinical trial science and operation to make clinical trials better, faster, cheaper. They are very inefficient in this country now, and this is attacking the IRB problem, the contracting problem, the clinical trial design problem, the adaptive trial design problem, the biostatistics problem, all of these things that apply to all different diseases. I want to just touch just for a minute on the international collaborations that we do. Translational science is a global enterprise. We all have the same problems. So we are big collaborators at NCATS. And so a number of years ago, we teamed up with our partners in Australia and Europe, and that is now extended, I'm glad to say, to England and to you. And we're really delighted. You're our first member in Latin South America, and this is a program which is advancing mainly the preclinical part of translation through global collaboration. We also have, if I can, I just want to mention that this these fundamental characteristics of a translational scientist, that double helix that I showed you a minute ago, was in fact a product of translation together. It came from a collaboration among global collaborators when we realized we all had the same issues, and so we wrote this together. And we all found that the same problems exist in the U.S. as they do in Canada and Europe and Japan and Australia, and now we know in Brazil as well. Pollyanna mentioned that I was the chair. That's me in the red circle there. It's a meeting in Paris in 2017 of the International Rare Diseases Research Consortium. I would urge you to look into this. One of my disappointments, actually, of my 3 years as chair is that I was not able to get very many rare disease organizations from Latin America involved. There is one Trans-Iberian Alliance for Rare Diseases. We're having a little bit of success there now, but this is now a collaboration of about 60 organizations in 22 countries on virtually every continent, and is doing amazing work to share resources, share knowledge in rare diseases, and make progress faster. Virtually every one of the RDCRN, Rare Disease Clinical Research Network, that I mentioned a minute ago, have international sites. None of them are in Brazil that I'm aware of, but every one of these has international collaborators. Okay, lastly, just the last couple of minutes, I want to tell you about how we applied all of this to COVID-19. First of all, we took a distinctly different approach to COVID-19 compared to our partners at NIH, Tony Fauci's institute, or all the other institutes at the NIH. We took a translational science approach to this, and one of those is that it's very collaborative and it's open. And I just want to give you a couple of examples of this. Very early on, many people were trying to do drug repurposing for COVID-19, but nobody was sharing the data. Nobody was sharing the complete data. And all of you know that you make progress much faster if people share complete data sets from these kinds of experiments, and including sharing the negative data, the compounds that didn't work. Because, of course, for molecular modeling and computational chemistry, the negatives are just as important as the positives. And so we created this open data portal in a week, literally in a week, and screened our collection of all approved drugs approved around the world, thousands of medicines and lots of other potential drug candidates across every one of these, and made all the data public. Positive data, negative data that had no effect, that's what the black means, and you can sort this in any way you want. Detailed protocols of every assay we did, and we did this immediately upon getting the results. We did not wait for the publications. We put them right on our website, and we've been glad to see that a number of organizations internationally have decided to put data in the portal too. And we really hope this is going to be something which continues post-COVID. Another thing we did early on was to create a mobile application, a mobile app that you could use on your phone, something called Cure ID, which allowed clinicians around the world to share their experiences of using existing drugs to treat COVID-19. We had created this together with the Food and Drug Administration, with FDA, right before COVID started, for repurposing mainly for neglected tropical diseases, and allow clinicians to share their experience treating those. But of course, in the early days of COVID, particularly before there was any clinical trial data, this was absolutely critical, and allowing clinicians all over the world in real time to share data with all the drugs that were used at the time. We also pivoted the tissue chip platform, which I mentioned to you, to COVID-19. One of these happened to be a lung chip, so we were able to pivot the lung chip platform, which had been developed for influenza, to studying the pathogenesis of SARS-CoV-2 infection and potential treatments, and that was a very useful platform for identifying repurposed candidate drugs in a much more physiologically relevant fashion. We created on the informatics clinical side an unprecedented electronic health record enclave. If you think about what we would have loved to have had early on in the pandemic, we would have loved to have known, who are these people? What sex are they? What race are they? How old are they? Are they children? Are they older people? What are their comorbidities? Do they have diabetes or hypertension, or what do they have? We had no way in this country to identify that. So we created it over the course of about 3 months, working with over 1,000 people across the CTSA program, investigators, and the informatics people at NCATS. We created this COVID collaborative. It now has over 1 million COVID positive patients in it, with records going back 2 years and new records being brought in every couple of weeks. Total number of patients is over 4 million. 1 million of those have COVID-19, and hundreds of projects from investigators all over the world who are using these data. And any of you could apply and get access to these data and ask whatever questions you want about COVID. Keep in mind, these are all U.S. sites so far, so that the data may not be directly applicable to those in Brazil, but I hope still useful. So what makes this special? Certainly the scale is one of them, but it's not just that we have a lot of patients, it's that they come from sites all over the country, rural, urban sites, all kinds of different sites. Over 5 billion rows of data, just think about that. And the demographics, you can't probably see this, but the demographics mirror perfectly the US population, which is very different from other more convenient samples, if you will. We also turned our clinical trial platform into this problem, and one of the ones we're doing is on immunomodulators for cytokine storm. Part of this is going through the active collaborative, active public-private partnership, if you're familiar with that. This is an adaptive master protocol trial of 3 immunomodulators work in 3 completely different ways to try to tamp down the immune overreaction that often kills people in COVID. And I want to emphasize here that though we started in the U.S., there are 4 sites currently being activated in Brazil, and there are 5 already activated in Peru and 3 in Argentina, and we're working on Mexico as well. I thought somebody might ask, where are the sites in Brazil? Here they are. And I don't know where these are, but you undoubtedly would know the cities that these are all in. They're all supposed to be activated this week or next and start recruiting patients, and those are the principal investigators, if you happen to know them. We also have been doing what we hope will be the definitive convalescent plasma trials. Many trials, as you know, have been done on convalescent plasma. One of the best ones actually done in Venezuela, but not good ones in the United States. And so we're well on to these slides, a little outdated, to having about 2,000 people who have been treated with well-titered convalescent plasma to figure out, does convalescent plasma work or not? And if it does work, in whom? So we should be able to tell that too. Okay, so what have we learned? What have I learned? And this is a little bit of editorialization from this experience. What I take away from this is that the operational problems, remember I gave you that list of scientific and operational problems at the beginning. The scientific problems remain, because they're going to be hard. But the operational problems, the lack of cooperation, the concerns about IP, the credit issues, all of those things evaporated overnight. And it showed that it is possible to go from fundamental discovery to therapeutics and vaccines much more quickly than has historically occurred. We know that because we just did it. We've seen this. So why? And I think this is because of the feeling of urgency. For the first time in my professional lifetime, everybody in the research ecosystem felt this urgency. Patients, and you all know this, patients feel this urgency every single day. Patients and their families feel that urgency. But researchers, public health officials do not, for the most part. And they prioritize other things. Now, all of a sudden, everybody had this urgency. That resulted in a recalculation of what people were willing to do, risk-benefit, based on that urgency. What then proactively, independently happened was I started hearing from people who wanted to share. I started getting phone calls for the first time ever. I want to work together with as many people as I can. People understood that the way to make headway was to work together. I didn't have to tell them that for the first time in my career. So there was a huge amount of proactive collaboration in the public sector, in the private sector, between the public sector and the private sector, from a desire to share. Now, when you think about this is potentially, and I emphasize the word potentially, a positively self-reinforcing cycle for the research ecosystem. Why? Because the increased productivity that we saw that came from the increased efficiencies gives a lot more credit to go around. So the concern that everybody has is, oh, if I share, oh, if I collaborate, I'll have to share credit. Well, if your productivity is 10 times what it was before, if you have to share credit with a couple of people, your individual productivity is still going up threefold or fivefold. So that has the potential to increase return on investment for everybody. But we're well aware in this country, and I'm assuming it's this way in Brazil, that the conditions that created this, the regulatory policy exemptions, the funding, all kinds of things will not, and the urgency will not continue without proactive steps by all of us in universities and industries, government sectors. We're trying to do that in the United States now, but we'll see whether we actually can keep up the learnings when COVID goes away, which God willing it will soon. Will we actually go back to the old system? I hope not. That would be a tragedy. If we actually learn something from this and the systems changes, that would be a suitable a suitable testament to all of the millions of people who have died in this pandemic. So that's the end of my comments. I'll just leave you with the translational spectrum as we diagram it here. You notice the patient's right in the middle here. We diagram this as a circular process because it is a circular process. It's not a linear process. It can go clockwise or counterclockwise. And I'd be glad to, as Pollyanna said, because we don't have time for questions, I would be delighted to answer any questions you have. All you have to do is send them to Pollyanna, and she will share them with me, and we will answer them via email, and that will allow you to ask them in Portuguese, which my Portuguese remains limited. But I would also urge you to connect with us on our website, Facebook, Twitter, YouTube, newsletter, etc. So, Pollyanna, back to you.
Thank you very much, Dr. Austin. It was an amazing and enlightening presentation. It's really wonderful to know about your initiatives there with COVID and also in NCATS in general. And I think it's vital for the translation research community to shorten the value of death in medical research with partnerships and learning from our partners' experiences. And it was just like really interesting for us listening and learning from you. So I am really sorry, as we told to the audience before, we are not going to have time now for questions, but we are going to reach out later and answer your questions further. And I just would like to to give the floor to our director, Dr. Carlos Morel, who is going to make some final remarks before we close up the session.
Thanks,
So
Poliana.
the floor is yours, Dr. Morel.
Thanks, Poliana. Chris, thanks us so much for this marvelous talk. I think we learned a lot, and you could compress in less than an hour a lot of information and perspective. It was really stimulating. And if we were already committed to collaborate with you, now this has increased fivefold.
Yay.
So I know you have a heavy agenda today, but really would like to thank you very much. And this was just the beginning. I think you are going to have a lot of connections, a lot of demand, and a lot of push for collaboration partnerships. Thanks a lot.
Perfect. Really look forward to it.
Yeah,
okay.
Perfect. Really thank you, Dr. Morel and Dr. Austin. And to conclude, I just would like to thank you again, Dr. Austin, and thank you for all the audience that is with us right now. And I would like also to invite all of you to our next session that's gonna happen on April 29th. So feel free to reach out and contact us through our channel on YouTube or email, and we are gonna keep in touch. So thank you, and see you next time. Thank you very much.
Right.
Thank you.
Bye.
Bye-bye.